Okay, welcome to Oceanography. This is week 12 or week 13. We've got two topics to cover this week. They're related topics that concern ocean productivity. And we're going to start out with a fairly brief overview of chapter 14, which is primary production. This is from Garrison, and there'll be a 10-point quiz on this. And then I'm going to try to get through most of that material, well, all of that material today. And then I want to start on the second concept that I'm going to present, which is nutrients from the sea for human beings. And specifically, I want to talk in detail around fisheries and fishing management and theories about how to manage fishing and overfishing and a lot of problems that stem from that. But I think to get an understanding of that and to fully appreciate the concepts that come out in this Nutrients from the Sea lecture, we need to make sure that we have some basic concepts about ocean primary production. And so that's what I'm going to do here for the first portion of this lecture today. And then hopefully I'll get done in time to open up the lecture for Thursday. In the beginning of the lecture on nutrients from the sea. Now, both of these lecture materials have been posted into the module. The quiz on nutrients from the sea is posted, and then there's a 10-question quiz that will cover material from the lecture material that I'm going to talk about now, as well as the readings in Garrison chapter 14. Okay. Before I start any sort of housekeeping details, you all had to hand in your milestone 3 assignments, and looking at across sort of briefly on that, there was a range of responses. Some teams did a super job. I was really impressed with sort of organizing, and although not all the parts were perfectly finished, there was a clear sense of the structure of the report and how the different components, the different portions of the report that were done by the various team members fit into an overall structure. So some of the teams did an excellent job on that. Other teams, not so much. There was a lot of sort of several teams where the product fell pretty far short of what we were thinking about a rough draft should be. And so we'll be getting back to you with that opportunity to try to improve that. But we're not going to do the same sort of revision that we did on milestones 1 and 2 because the revision is going to be the milestone for your final product. But we will give you a chance to confer with Carrie or me about your milestone 3 assignment, and hopefully you'll be able to use that information to improve the final product. So any questions about where we're at with the team project, the class project, or other material before I get started on the lecture? Any questions at all, please? Okay, well, I'm not hearing any questions, so I want to talk about material from chapter 14, which concerns primary production in the ocean. And the key concepts are primary production is the synthesis of organic material from inorganic substances. principally carbon from carbon dioxide, but also other nutrients, key nutrients are needed to complete this synthesis. And the primary production in the ocean is a source of roughly half, a little bit less than half of the total oxygen production on the planet comes from primary production in the ocean, which is an impressive number, but you have to consider that the size of the ocean, you know, the 2/3, a little bit more than 2/3 of the ocean of the Earth's surface are covered by water. So with half of the oxygen production, half of the primary production happening, that suggests that there's a lower level of overall primary production, overall level of per unit of Earth's surface. Most of the primary producers in the ocean are planktonic. That means that they drift freely with the currents. They have no ability or very weak ability to swim independently of the direction of the water. So planktonic organisms are all organisms that are at the mercy of ocean currents. There are, however, important vertical migrations that take place among the zooplankton, but phytoplankton are the primary producers, and they're confined to the upper layers of the ocean. The most productive producers are the cyanobacteria, and production is measured in the amount of carbon fixed per individual. They operate on what's called a microbial loop, which I'll explain in a little bit here. Not all primary producers are free-living, free-drifting plankton, because there are attached seaweeds, seagrasses, mangroves that are important components of the marine ecosystem and are contributors, locally at least, to overall primary production. And there is also chemosynthetic, chemoautotrophic organisms, microbes that live in the ocean. They can be free-living or they can be symbiotic within other larger organisms, and that's something we discussed previously and that we'll be returning to when we look more at chemosynthesis next week. Primary producers synthesize organic material. Phytoplankton are responsible for something over about 96% of the ocean's carbohydrate production comes from these phytoplanktons. They're typically measured in grams of carbon per square meter of ocean surface per year. So grams of carbon per square meter of ocean surface per year is the metric of ocean primary production. And the source of the carbon in fixing this carbon is carbon dioxide. Carbon dioxide is almost never a limiting nutrient in the oceans because it's freely available from its carbon dioxide, and its other products, bicarbonate, are highly soluble in ocean waters. So other nutrients are typically what are the limiting factor for primary production. This sort of a diagram here. This is sort of encapsulating what I was talking about. So we have 1 square meter of ocean surface, and that square meter is receiving sunlight depending on latitude and season. And that availability of sunlight, of course, is one of the principal limiting factors. So that using the energy from the sun in photosynthetic metabolic pathways, primary producers, planktonic producers, are able to take carbon from carbon dioxide and to fix it as glucose. And typically, ocean primary productivity in the water column produces about 120 grams of carbon into molecules of glucose every year. So about 120 grams of carbon per square meter per year is a sort of a typical metric of ocean primary production, averaged globally. Some areas will have much higher levels of production, and other areas of the ocean tend to be less productive. For example, the tropical, equatorial tropical ocean, open ocean areas tend to have low primary production because there's a limit to the availability of nutrients. And so they're often considered ocean deserts, whereas coastal areas where there's more available primary nutrients, they tend to have high levels of primary production. And seasonally, the highest levels of production occur in the high latitudes, where during summer months, depending on north or south hemisphere, there can be light for many hours of the day. So the availability of sunlight, which is of course one of the things that drives primary production, can be very high in the high latitudes, and therefore seasonally you get high levels of production. So we can think about this primary production occurring in the surface ocean where there are phytoplankton. So the phytoplankton are confined to the surface ocean, but the zooplankton, the multicellular organisms that eat the phytoplankton, the primary consumers of the primary producers, they are actually able to migrate vertically. And so the combination of particulate organic carbon, the dead bodies, the waste products of the phytoplankton and their zooplankton consumers sink through the water column. So that's a loss, or that's a way in which fixed carbon from primary productions in the ocean surface can reach the sea floor. And of course, this is going to be limited by how deep the water is. The deeper the water, the less of this carbon reaches the sea floor. And then the migration of zooplankton, which is a massive movement of biomass that happens on a daily basis. So typically during the night, zooplankton will come up into the surface waters and feed on the phytoplankton, and during the day they migrate back down again into deep water to avoid predators. So predators seek out zooplankton according to their visual, using visual cues, and so to avoid that, zooplankton tend to migrate up and down. And this is something that was discussed in that mid-ocean, mid-water quiz and video that you all looked at. And so the movement of these organisms is a pump that moves water, that allows carbon to reach the sea floor, and also allows carbon dioxide to be re-emitted into the atmosphere. And the diurnal migration takes place, diurnal vertical migration, and this is part of what's called the deep scattering layer, because scientists first realized that this was happening when they detected acoustically the presence of vast numbers of organisms that were moving up and down during the course of a 24-hour period. And this is, again, the material that was covered in that. So the deep scattering layer, is a high signal, so that depending on depth, there are different layers in this, and the basic idea is that the organisms that comprise the deep scattering layer move from the depths to the surface and then back down again, according to a 24-hour cycle. And these animals are detectable typically because they contain tiny bubbles of air, swim bladders in the fishes, and other sort of pockets of air or pockets of oil that can be detected using acoustic energy. And so the deep scattering layer is a function of this migration and is a part of the biological pump. So plankton collection methods, depending on the size of the plankton used, the different fish mesh sizes will be used to collect different species of plankton. And let's see. So the typical means for plankton connection is what's called a plankton net. And This is typically a sort of a net ring, and usually the size of this opening is fixed at 1 square meter or some a fraction of a square meter, so that by towing this through the water at a known speed, we can tell, or scientists can estimate, the volume of water that they're sampling over time and get some kind of an idea of the density or the average density of the animals that they catch. So they take this opening and use that as a denominator in calculating, and then they know by recording the amount of time that they're moving the net through the water, they can get a rough estimate on the volume of water that they've sampled. Of course, a lot of these planktonic organisms are able to swim actively enough to avoid the net, so the size of the catch is a biased estimate towards animals that are less able to escape. And then the size of the net fabric is going to determine what is caught in the net versus what is able to pass through. So for collecting phytoplankton, you would have to have a net size that's quite small. Most of the phytoplankton are microscopic in size. So phytoplankton are autotrophic. They generate glucose by photosynthesis, and roughly half of the food generated by photosynthesis on Earth is produced by phytoplankton. Picoplankton are plankton that tend to be between 0.2 and 2 micrometers across, whereas more common phytoplankton are in the order of 5 microns up to about 50 microns. Cyanobacteria are also in the picoplankton range. They tend to be microscopic in size. Okay. So the phytoplankton are the base of the food chain, and I've talked about this concept of a food chain. So we have the primary producers, and they're taking dissolved inorganic material, nutrients and inorganic, they're combining these using energy from the sun and photosynthesis. They're combining these dissolved inorganic nutrients to make new organic material. Examples of different phytoplankton include diatoms, dinoflagellates, coccolithophores, and I'll talk a little bit about each of those groups. And these are in turn consumed by zooplankton. Copepod here, this is representing a copepod, and the copepods and zooplankton are eaten by small fish, which are eaten by large fish. And that's sort of the typical food chain that we think of. And so the large fish are, and all of these have a return loop, so that when these organisms die or are consumed, they return dissolved organic material and inorganic nutrients back into the water column that can resume this cycle. So the decomposition wastes contribute back to the dissolved inorganic material which the phytoplankton are using. The microbial loop refers to cyanobacteria, which are able to utilize this dissolved inorganic material. And that tends to be in different forms of cyanobacteria, and some of them consume and some of them break down. But this is a highly productive loop, and depending on the efficiency of the traditional food chain, the microbial loop can be greater or can account for more or less of this total inorganic nutrients. And so one of the concerns, if you were to take a typical food chain like this and disrupt it, for example, disrupt it by removing fish from this food chain, well, the decomposition products of the zooplankton would be returned more readily into the water column, and so that the damaging or reducing the effectiveness of the traditional trophic loop is going to increase the amount of production and consumption represented by the microbial loop. And this is a particular concern in parts of the ocean where overfishing, pollution, and other processes have damaged or limited the traditional food chain or food web. And so we'll see when we look at fisheries and the effects of overexploitation in fisheries that the increase in productivity by the microbial loop has become increasingly important in the global ocean to the detriment of biodiversity and overall ecosystem health. Okay, so just a few words on the different types of phytoplankton. Diatoms, which are among the phytoplankton the most productive, they're characterized by a rigid cell wall of silica, and most of them float. Others can lie on the bottom in shallow water. The glass silicate cell walls come in typically in two sections, one fitting inside the other, and that is what gives rise to this diatom, so two atoms, not really atoms, but two sections of frustules, the rigid cells that fit together. And there's concern about the overall abundance of diatoms. So let me just switch away from this slide to a brief video from Wikipedia.
Diatoms are one of the most abundant types of marine phytoplankton, but a new 15-year-long NASA study reveals global populations have declined. Diatoms, like all phytoplankton, have chlorophyll, the same photosynthesizing pigment as plants. They occupy the surface of the ocean where they harvest light from the sun. In large numbers, diatoms form colorful swirling blooms that can be seen from space. Scientists use NASA satellite data, water samples, and computer modeling to determine how diatom populations have varied. This data visualization shows changes from 1998 to 2012. According to the study, significant decreases in population, shown here in red, are mainly in the northern hemisphere. Scientists link this to a shift in the availability of nutrients.
Diatoms rely on nutrients such as nitrate, silicate, and iron to reach the surface layer where they live. What our study shows is that the availability of these nutrients has changed due to the way they cycle within the water column.
Diatoms occupy the surface area of the ocean called the mixed layer. Nutrients collect on the ocean floor and are cycled up to this layer. Various physical forces can cause the depth of the mixed layer to become shallower, so that fewer nutrients reach the diatoms. Without these nutrients, their populations decline. This map shows areas on the globe where the depth of the mixed layer shallowed.
It's hard to pinpoint exactly why this change have happened. Things like wind, circulation, and temperature can affect the way these nutrients are brought to the surface layer. We hope a longer study can yield more information on whether these changes are in fact a trend or variability.
Okay, so that was just a brief video looking at diatoms and linking together some of the oceanic processes that we've been talking about, the mixed zone, the availability of nutrients, the recycling of material from depths to the surface. And so we can see once again that each component of the system, relies on other ocean processes. And so concerns for changes in the typical conditions of the ocean, seasonal changes, circulation changes, may be expressed as changes in the overall abundance of very important marine organisms. And so, while there isn't conclusive findings that diatom production has decreased, there is some indication that may be a matter of concern. Another species which interacts with human beings are dinoflagellates. And dinoflagellates account for about 30% of overall phytoplankton abundances. They're unlike they have a flagella, and they're able to move using these flagella and steer themselves into areas of higher production. They're commonly bioluminescent, and harmful algal blooms such as red tides or brown tides are often caused by overproduction of diatoms. So one of the things that may contribute to this is if there's a oversupply of nutrients needed by the diatoms, and this can produce a huge increase and a very rapid increase in population size. And this is something that we're familiar with in Florida because we've been through a series of red tide type events. Red tide is a particular tide caused by a diatom called Karenia breva. And Karenia breva seems to bloom episodically along the Florida coast. And there's not totally certain why this occurs, but it's thought that failure to process human waste and sewage process products and other sources of organic pollution may contribute to the abundance of, to the occurrence of red tide events. And they're a concern because they produce toxins which can be harmful to fish, they can be harmful to human beings, and they're unsightly. And so Florida economy, which depends so heavily on marine resources and the marine environment, can be jeopardized by the overoccurrence of harmful algal blooms, HABs. And the third species of, or a group of phytoplankton that we'll talk about are the coccolithophores. And they have, unlike the diatoms, they have discs of calcium carbonate. They account for about 25% of overall phytoplankton abundance. And their skeletal material being made of calcium carbonate is subject or vulnerable to changes in ocean pH, to ocean acidity. And so with the increase in carbon dioxide, just dissolved carbon dioxide, and the increase in acidity, decrease in pH in the ocean, which has been observed recently due to increasing carbon dioxide levels in the ocean, the ocean waters have become more acidic, and there's gathering evidence that this may be impacting coccolithophore abundances globally. And there have been various studies where scientists have recovered coccolithophores that show some evidence of dissolution in their calcareous tests. And so there's concern that, and also when the acidity drops, it's more difficult, it takes more metabolic energy for calcareous plankton to precipitate the calcium carbonate, which they use to make their shells. And so that can reduce the overall population levels and productivity of the coccolithophores. So both with diatoms, dinoflagellates, and coccolithophores, there's a possibility for human activity to change the abundance and the health and the outcome of these populations, to the extent that these are matters for concern as we observe changes in ocean chemistry and ocean health occurring with climate change and with other human influences. So let's talk a little bit more about the factors that control the production and availability of phytoplankton. So the lack of nutrients and light are the limiting factors for primary production. As I said earlier, the availability of carbon dioxide or carbon is very rarely a limit in the ocean due to the high solubility of carbon dioxide within the ocean waters. So phytoplankton balloons can cause a lack of nutrients for other organisms. Nutrients are commonly available in colder polar waters. There's light availability. So IronX refers to as an experiment that tried to enhance primary production by seeding the ocean with iron. Iron can be a limiting factor and very often is a limiting factor for phytoplankton production. So one of the ideas was that we could increase artificially plankton production by artificially supplying one of the limiting nutrients, namely iron. And so during the early 2000s, there were a number of experiments undertaken where scientists would seed the oceans with available iron, ferrous iron, that would then enhance production by supplying a limiting nutrient. The results from this were not as, and the idea was that maybe if we could increase primary production in plankton, we could increase their ability to remove some carbon dioxide from the atmosphere, and this might be conceivably a sink for excess carbon dioxide in the atmosphere. Unfortunately, and the idea was that you could have ships sailing around scattering iron, and that would induce increased primary production, and that would reduce carbon dioxide. In fact, it turns out that this probably will not work. There are other factors that limit iron production and primary production. And so simply providing extra iron does not appear to significantly increase overall primary production. The availability of light tends to limit photosynthesis below about 100 meters. That's going to vary differently in different parts of the ocean. If there's too much light, that tends to overwhelm photosynthetic chemistry. So typically, you don't find phytoplankton in the very surface waters of the ocean. They typically don't float right at the ocean surface. They like to be a few centimeters or a few meters below the ocean surface, where they're protected from excess light. And the color of the light matters. And of course, the color of the light matters because different wavelengths of light are absorbed with different efficiencies by ocean water. And so here we have a diagram that shows what happens to white light, sunlight, as it passes through water. Of course, sunlight ranges from really short wave lengths, about 300 nanometer, all the way out to red light, which is about 800 nanometers in size. So we go from violet to red, the typical rainbow. But not all of these light wavelengths, not all of these colors of light penetrate equally through seawater. And so red light, longer wavelengths tend to be extinguished or absorbed very quickly in seawater. So anybody who's ever gone snorkeling or skin diving or scuba diving has noticed that as you descend, within a couple of meters from the surface, reds disappear. Because there's no longer any red light available to reflect off of red pigmented surfaces. So red fish that appear red in air will appear brown or dark colored at depth because there's no available light. The light wavelengths that have the greatest ability to penetrate the ocean are green and blue. Blue light goes all the way down to available light all the way down to about 250 meters. This is for clear oceanic conditions. Okay. Green light also penetrates quite deeply, and you'll recall that accessory pigments were required for organisms that live deep in the water column, where they're limited by the availability of red light. So you tend to get red-colored organisms, or red-colored pigmented organisms that can absorb green light. They occur at greater depths. And this is another way of looking at this same concept. This is light extinction by different wavelengths. And what this is illustrating are differences in water quality and how that produces different light conditions at different depths. So on the x-axis in both of these diagrams, we have the wavelength of light. And this wavelength would be from violet, short wavelength, all the way out to red. Okay. And then these lines here are isobaths, or isobaths for light penetration. So here you can see that red light disappears very quickly in the open water. So this is the irradiance. This is measuring the intensity of light per square centimeter at for different wavelengths. And so below 10 meters, there's almost no of the longer wavelength light available. So all that light is absorbed. And and then this progressively, the distribution of available light decreases, similar to what you're seeing in the graph on the upper left here. This is sort of inverting that diagram, and you can see that it's the light in the range of about 450 to 500 nanometers that penetrates the deepest. Okay, so this is for clear ocean water. And this is from the Sargasso Sea, which is basically the Atlantic Ocean in the area of the Bahamas. This is an area where there's very low primary production, limited nutrients, and as a consequence, the water tends to be very clear. So clear water is usually an indication of lack of nutrients and lack of primary production. So that's in the Sargasso Sea. If we go to the Baltic Sea, the Baltic Sea is the area between sort of the Scandinavia, between Russia and Scandinavia, between Europe and Scandinavia to the east. That's the Baltic Sea, and that's a relatively shallow sea with high nutrient loads due to runoff from rivers all around. And here you can see that you have the same sort of diagram and extinction of light, but if you look at how deep the light is penetrating, you can see that the maximum penetration of light cuts out at about 20 meters. Plankton living in the Sargasso Sea would be able to absorb green light all the way down to 100 meters, whereas in the Baltic Sea, below about 20 meters, there wouldn't be, they would start to be limiting available light. So, photosynthetically active radiation is the light in the wavelengths usable for primary producers. And this ranges from about 300 to about 700 nanometers, basically the wavelength of visible light. And you can see by looking at this that different types of organisms use light in different types of settings. So the red algae, these are algae with accessory pigments, they have red color, and that red color allows them to absorb green and blue light. Diatoms, green algae tend to absorb light in this blue range here, all organisms like red light, but red light tends to be relatively limited. Okay, so this is called photosynthetically active radiation, PAR, and that is the indication of the spectrum of light which can be used by primary producers. So 300 to 700 nanometers is the range of photosynthetically active radiation, PAR. Okay. Why does this matter, and how can we use this? We can look at this in various different ways. And the light extinction in the ocean is controlled by a simple exponential decay formula. And so we can measure I, which is the intensity of light, at a depth Z relative to the availability of light at the surface, times the natural algorithm raised to the negative K times Z. So Z here is depth, and negative K is an exponent that measures the ability of light to be transmitted through water. Okay? And so, here is a diagram of sort of the availability of light, and this is being measured in microwatts per square centimeter, light intensity, and this is light intensity on a logarithmic scale. And so this is saying that the availability of light for phytoplankton growth, you have to have at least 10 to the second to 10 to the third microwatts of energy per square centimeter to support phytoplankton growth. Okay. So this is going to suggest that in clear coastal water, you could get productivity down to about 20 meters, and that's what we're seeing in that graph that I showed you from the Sargasso Sea. The clearest ocean water, represented by this graph here, would be the in the clearest ocean water, you could get available sufficient light to support phytoplankton growth down to something like 200 meters. Okay. And then, some other sort of interesting factors that have to do with light intensity. Moonlight, it turns out, will actually penetrate water. So moonlight would be visible down to this kind of range. Color vision is limited here. The ability of phototaxis, in other words, to orient towards high and low light, sort of cuts out at about, in clear ocean water, at about 800 meters. And the detection limit of human beings is down here. So the human eye and deep sea fishes would actually be able to detect some difference in light all the way down to about 1,000 meters. So I've never done it, but I've heard reported that if you sit on the bottom in a submarine and turn out all the lights, that gradually over time your eyes will adapt and you'll be able to make out vague shapes, even at significant depths like 400, 500, 600 meters. So this is sort of illustrating how the extinction of light is determined by this, controlled by this simple formula. Now I'm going to show you, here are some results that I obtained from a coral reef in offshore Texas. This is the Flower Garden Banks. This is a national marine sanctuary. It's a very productive reef. It's one of the most productive sort of coral reef settings that we have in the United States. It's far enough offshore so it doesn't get a lot of traffic, a lot of use, and so the health of the Flower Garden Banks has been quite good over time. And the water is, because it's offshore, tends to be quite clear. So I conducted an experiment where we deployed a light sensor, that's this sort of light bulb looking device, and that was connected to a CTD. Remember I talked about CTDs earlier. And so we can see here, this goes back quite a while, this is sort of looking at a series of days. I guess this is about 5 days. And you can see PAR, photosynthetically active radiation. You can see that increasing during each day. And so, and going to very near, close to 0 each night. So this is the daily cycle of night, light, and darkness. You can also see, interestingly enough, the dissolved oxygen component. So this is the availability, the measured amount of oxygen in the water. And you can see that the amount of oxygen in the water very closely tracks the amount of light in the water. And what that's measuring is the productivity of the primary production of the organisms within the water column. Now, a lot of, at a coral reef, a lot of the primary production is actually being conducted by the photosynthetic symbionts of the corals. So here we can see how the availability of light controls that. All right, well, what does that say about how variable this is? Because you can see, if you look at the light here, that it's not constant. It goes up and down, and there are a variety of things that could be determining that. One of those is the availability of light, cloud cover. So if it was a cloudy day, you'd be getting less light, and you would expect to see less light at the bottom. And so one of the questions that I had was, can we take out the effect of clouds and clarity of the air, and just look at how that's varying in the water. So what I did was to use this, taking this device here. I had a device that was measuring light at the bottom of the ocean, and I placed a similar device to measure light up on an oil platform. So there was a disused oil platform about a mile or so from the study site in the flower gardens. And so I could compare the irradiance of light on the platform with the irradiance of light at the bottom in the flower gardens. And the water depth there was about 30 meters. So here you can see, the irradiance, and this is measured in moles per square meter per hour, a somewhat different unit, but that's a common unit there. And you can see this is over the course of, almost a year, and then the light goes up and down seasonally. In December, we had some storm events and things that made that difficult to measure. So this is the basic record of irradiance, incoming sunlight on the oil platform. And here is the same measure of irradiance for the same period on the bottom at the West Flower Garden banks. And so this is the light availability, photosynthetically available radiation dose measured in moles per hour per square meter, the same unit. And you can see that this is going up and down seasonally. And then there's also a daily pattern built into that. If you take the, if I compare directly the radiance I measured at the bottom with the radiance at the surface, that takes out the effect of cloud cover. And so we can calculate the value for K, the attenuation of light in the water column, as a function of changes in the water, the availability of the cloudiness of the water, the amount of primary production in the water. And you can see generally from this graph that attenuation increases, negative K becomes a greater value, so light is more attenuated during the summer months. And that's kind of what we'd expect. We'd expect more primary production during the summer months and reduced primary production during the winter months. So that's an example of using measurement of light to understand the way in which primary production is affecting availability of oxygen, clarity of the water, and overall primary production. And so at any given time, at any different point, you have a depth, compensation depth, where the amount of light, the available light, is going to allow sufficient respiration or sufficient availability of metabolic respiratory products to allow plankton to survive. And so, below this compensation depth, there's not enough light for photosynthesizers, for primary producers to compensate for their metabolic needs. And so below the compensation depth, there's essentially no phytoplankton can't survive. And then as you go up in the water column, you see that photosynthesis is inhibited toward the surface because there's too much light. And so the depth of the greatest productivity is going to be somewhere in the middle of this range. Growth productivity and net productivity. So net productivity is the productivity in excess of the metabolic requirements for phytoplanktons. Now, this graph is drawn, you can sort of see here that they're suggesting that this graph suggests that the compensation depth should be something greater than 200 meters. But we know from the previous data that I showed you that the compensation depth in different water bodies, different oceans, different ocean types could be much less. So that in, coastal waters with high turbidity, this compensation depth would be much farther up in the water column than we're seeing here. Okay, so that's a view of that primary production. I want to say further that light compensation points. So this is sun grown, shade grown, light intensity, and different factors. Primary producers face a metabolic cost, and they need to produce enough carbon through photosynthesis to grow. And so the light compensation points, are going to occur at some. This is sort of measuring the compensation depth. So this is the compensation depth shown as a measure of light intensity versus carbon dioxide uptake. And so greatest saturation is going to be, in this range here. And the phytoplankton values are going to, phytoplankton productivity is going to vary with local conditions. And so in the tropical ocean, you get, the seasonal patterns don't matter nearly as much. So you can see that, this is sort of a conceptual diagram, more and less, and summer doesn't particularly determine what so water temperature is constant, nutrients are constant, and the population of phytoplankton are relatively constant. In the North Pacific, we can see a much stronger, so this would be up in here somewhere. In the North Pacific, we can see a much stronger seasonal variation. So that in the North Pacific, we're going to get more nutrients in the wintertime and less nutrients in the summer. Well, what's causing this? This nutrients to be more abundant in the winter months and less abundant in the summer months? Well, that's the response of the phytoplankton growth. So phytoplankton grow in response to the availability of light, and the increase in the phytoplankton populations causes a decrease in the overall availability of nutrients. So this is a North Pacific sort of temporal ocean response, and with the ocean temperature varying on a seasonal basis here. It's different in the polar region. So this is polar areas in the Baltic Sea, the Bering Strait, so forth up here. If we look here, the temperatures are always pretty cold. It never really warms up. But in the summer months, you have very long days. So there's high availability of light, and that produces, so there tends to be high nutrient availability through most of the year. And then that can be slightly reduced by the seasonal spike in phytoplankton production. So phytoplankton productivity varies with local conditions and varies seasonally, and so we would expect very different sort of annual profiles in productivity depending on if we were in the tropical ocean, in the temperate North Pacific Ocean, or in a high latitude setting. Okay, well, I'm going to stop there with material on from chapter 14. That's as much as I really have time to present if we're going to get to other topics. But are there any questions over what I've talked about in the first portion of this lecture? Does anybody have any questions or interests in this? So if I were to ask you on in a quiz, for example, if I provided you with a depth and a surface intensity and asked you to calculate what the irradiance at some depth would be based on some value for K, would you be able to do that? Yes? No?
Most likely.
Okay. How would you approach that?
I would personally plug in the numbers given by the question into the equation and solve for, you were asking for K?
Right. If you're asking for negative K, or if it provided you with negative K, if I gave you a value of negative K and gave you a depth, you would just take negative K times the depth and multiply that by the irradiance at the surface, and that would give you the irradiance at depth, right?
Correct.
Correct. But suppose you just knew the irradiance at depth and the irradiance at the surface. If you took the natural logarithm then of that irradiance at depth, you'd be able to calculate what this K value is, wouldn't you?
Yes.
Yeah, so that would be the approach that you would use. So that might be something that shows up on the final exam, as a longer answer type question. Any other questions or thoughts about primary production before we go on to the next topic, which is nutrients from the seed?
Could you post a practice question of this?
Yes, sure I could. Yeah, I'll give you a practice question of that. And it won't be today, but I'll put it up tomorrow. Yeah, I'll show you how to do that.
I have a question about, well, a couple slides ago, there was a point that said diatoms have a greater compensation depth than dinoflagellates, but I'm confused. What's the difference? I think I missed that point. I'm a little confused on what the difference is, or is it a type of phytoplankton, or is it different?
Well, there are three main groups of phytoplankton. So, oops. Diatoms, and diatoms tend to be the most productive. They tend to occur sort of mid-ocean, sort of mid-photic zone depths. And dinoflagellates like to be closer to the surface, typically, and the coccolithophores range a little bit deeper. So, these are three groups, and they're classified according to what their body walls are made out of, and their ability to move, the presence of flagella, and whether they have tests that are made out of silicate versus tests that are made out of shells that are made out of calcium carbonate.
Got it. Thank you. I was only a little confused, but that clears things up. I figured it was the types of phytoplankton, but thank you.
Right. So yeah. So so an exam question would be, what are the three main types of primary producers in the ocean? Well, you'd say diatoms, dinoflagellates, and coccolithophores. And I hope that all of my students come out of oceanography being able to say that if it comes up in conversation, what are the major types of phytoplankton? You'll be able to say diatoms, coccolithophores, and dinoflagellates. And then, of course, cyanobacteria are also primary producers, but they tend to operate in a microbial loop separate from the main food web. Okay. Okay. Well, let's press on with the second topic here. We got another 20 minutes or so, and I can get started on this. So let me escape from this one. And close this one. This one. Oops. So I'm going to spend more time on this and go more deeply into this, because this is, something where a lot of the processes and the oceanographic principles that we've been discussing throughout the course kind of come together, because not only are we looking at, currents and winds and upwelling and those processes, we're looking also at primary production and the food web, biodiversity, marine communities. They all sort of come together in this when human beings get involved in their by extracting nutrients, food from the oceans. And so it's a good example a good opportunity to, to take a practical subject like fishing and fisheries and examine the ways in which oceanographic principles and processes contribute to that, and also the ways in which, the ocean kind of limits what we can get out of it, which is, in a lot of ways where we're at these days. So we'll start by saying that, human beings have depended on the sea for a very long time. This is not a recent phenomenon of, humans depending on fish. And a recent example of this, well, not all that recent, I think this discovery was in 2012 or 13, something like that, that archaeologists found a footprint, a human footprint on Calvert Island in British Columbia, which they were able to carbon date by examining material somehow or another in the footprint. And they calculated that this footprint was 13,000 years old. Well, 13,000 years, and this is in North America, that would put this, way back, just as human beings were first arriving in the North American and, well, the American continents. And so the concept is that human beings migrated across the Bering Sea Bridge when the glacial a maximum had occurred, and human beings were able to migrate that distance. And so, one of the questions has been, once people crossed that land bridge, did they disperse and colonize the interior of North America and eventually South America by crossing through the land and chasing land animals, hunting caribou and deer and other organisms, or did they migrate down the coast? And so, there's a variety of different concepts there. But this 13-year-old footprint was evidence that, at least some human populations would have migrated along the coastal area. And this makes a lot of sense because, coastal areas tend to be highly productive. We talked about that when we were looking at, tide pools and tidal communities. So kelp forests, oyster beds, populations of seals and other marine mammals, fish, of course, all would have been available for food products. And, so that there's, Native Americans did form agricultural communities, and there were some very large cities and civilizations in the interior of what's now the United States. But there were also very large populations around in coastal areas. And in Florida, we can find evidence of populations based on shell middens. And these are shells that Native Americans collected from highly productive ocean areas and ate for food. And these shell middens are huge stacks of shells, indicating a large population of people being supported by oysters and long periods of time when these populations were present. Now, one of the interesting things historically is that when the conquistadors, the Spanish explorers, came into the Gulf of Mexico, they were surprised by the health and the robust physical condition of the Native Americans that they encountered. And because these populations, these native communities had learned to extract maximum food from the oceans. Whereas actually the Spanish and the Portuguese explorers were not nearly as adapted to that. And so, a lot of the expeditions kind of came to grief because the explorers ran out of food and, literally starved to death in the middle of this oceanic abundance. There were abundant fish and abundant, other oysters and other foodstuffs they could have collected, but they hadn't culturally acquired the skill and the ability to do so. The ability to fish and the ability to extract nutrients from the ocean is something of long standing, but there is cultural dependencies on that. Okay, I'm going to we're going to talk about fishing and fish, but I wanted to take the opportunity to show a little video about the traditional methods of collecting fish. And this is going to be, let's see, I'll escape here, and I'll show this video here. So this is talking about the Mid-Hayden fisheries and the way in which Menhaden were collected. So let me make this full screen. And we'll play.
For countless generations, the Menhaden fishermen have been setting out to sea from Beaufort, North Carolina. The fish swim in schools of millions. They've been netted since Indian times for fish oil and fertilizer. Menhaden fishing never has been easy work and still isn't, even on Captain Bobby Martin's boat rigged with winches and pumps.
They don't exactly jump on this boat. We had to get down and work, get them on here, you know, but it's nowhere near like it used to be.
It used to be harder. That is something Captain Martin does not have to tell the three honored guests he has with him this trip. They know how it used to be. John Henry Pritchard knows. 50 years ago, the Menhaden boat on which he was working went down in Beaufort Harbor. 17 of his shipmates drowned that day or froze to death before anyone could get to them. John Henry Pritchard knows how it used to be.
On the back yonder, on that bridge, ice had frozen up a solid post of ice. About 4 of them back there, to them holes.
John Jones does too. And so does John Bell. He remembers.
And it got so rough, we had to let the fish go. And we tried to take our boats up.
This is how it used to be. They didn't have any hydraulic winches to pull their nets. They had strong arms. The nets weighed thousands of pounds. If the men did not pull together steadily, powerfully, fold by gathered fold of the net, all exactly at once, the catch might be lost. They might be lost overboard with the enormous slipping weight of what they held in their hands. To get themselves pulling together, to lift the nets, to lift their spirits, they sang.
You are so sweet. Bye-bye, bye-bye. Bye-bye, bye-bye, my darling child. I won't be home tomorrow.
Mike Luster of the North Carolina Maritime Museum had heard about the hauling chanties the old Menhaden men sang.
And I said, is there anyone around who remembers these songs? Who sang lead with these songs? He said, John Jones, and he lives right over here.
Mike Luster discovered that other retired Menhaden men were still to be found on shore. Before long, 13 of them were getting together most every Friday night, trading stories of how it used to be, and singing the shanties that got them through the hard, cold work at sea.
It would get so cold and the wind would swell and bust. You still had to pull. You had to carry your load. So you start singing them shanty songs, they give you spirit to go on. And that's the way it was. You had to learn how to pull. As you pull one hand, you catch the other hand to pull. You pull again, you throw it this way. Next time, you throw it that way. You keep it level. So you have a level place so you can have your balance. Keep your balance. And you put
Well, I'm going to stop there, but I'll put the link to this really interesting little video there. But I wanted to show it because it illustrates the way in which, let's see, I have to escape from there. That, the traditional work at sea, used to be very much a question of human power, of people pulling on lines and ropes, and sea shanties were a part of the way in which people organized their work around this very, very hard physical labor in a way that allowed them to maximally work together to keep their hauling and their maximum effort. And so that was an example of that. And the Menhaden fishery and the type of fishery that they were doing, this is a type of fishery called purse seining. And so purse seining, up until living memory, was something that was done by human power. And so, with large numbers of people needed and having to physically pull these nets, that was very much a limit on how much purse seining could be done, and how far offshore people could go, the kind of and how many men it took to operate a purse seine fishing. And so that was that really limited how much expansion of that method of fishing could take place. Well, that changed with technology. And one of the things that, one of the key inventions that really revolutionized fishing was the development of what's called the Puretic power block. And this is a type of winch which is operated by hydraulic power. So there's hydraulic lines that go up to this block. In this case, it's a block on the edge of a on a tackle here. And so instead of having people pulling on these nets, they can operate this machine and have the machine pull nets. And so the Puretic power block was introduced, I think, in probably the early 1960s. It was actually an invention of a person originally from Portugal, who came and operated fishing and invented this device for use in the sardine fisheries of California. And the sardine fishing was another traditional fishing. It's fishing a pelagic fish species, kind of like the Menhaden. So the sardine fishery really exploded as a consequence of people using this Puretic power block as a means for catching fish. And I'm going to switch here. I guess I've got about 10 minutes left, and I want to talk about the anchovy fishery of Chile and Peru, Peruvian anchovy, and use that as a way of illustrating how these different factors come together. So keeping in mind that in the late 1960s, early 1960s, this technological innovation came along and revolutionized the anchovy fishing. Okay, so but the use of the hydraulic Puretic power block was not the first time that human society had been strongly influenced by the very productive fishing grounds that are located off Peru and Chile. In the late 18th century, so there was, European society began to experience a limit in the availability of food because the agricultural production, which supported human society, came up against a nutrient limit. And the nutrient limit was the availability of phosphate and nitrate for use as agricultural fertilizer. So the farms, the agricultural production in Europe, had sort of gotten to its maximum capabilities. And no matter how much, the natural sort of fertilizer was to use manure from livestock, but even with maximum manuring, use of manure as fertilizer, the amount of yield per acre sort of had reached a plateau. It actually begun to decrease with decreasing soil health, and as a consequence, there were episodically massive famines that occurred throughout the 1800s. And so at various times, because of bad weather conditions or other problems, crops would fail. And when crops failed on a certain year, the human population would starve, literally. And so the ability of agricultural production sort of had hit this natural limit. And this nutrient limit and the availability of nutrients limiting a population is something that we've been talking about. We talked about it with phytoplankton. We're going to talk about it with fish. So it's the availability of available nutrients can limit populations. And here's an example where the availability of nitrogen fertilizer and phosphorus fertilizer to produce plant growth became a limiting factor for human populations in Europe. Well, one of the things that at least temporarily overcame that was the discovery of what are called the Guano Islands. And the Guano Islands are small rocky islands located off the coast of Chile and Peru, where there are huge populations of birds, and particularly the Gorai cormorant. And Gorai cormorant is a seabird. It eats and they like to build their nests colonially, and so they come back and they build these little nests, and the nests are built from their droppings, from their guano. And the climate conditions on these islands are essentially totally arid. It almost never rains. And so under conditions of aridity, these guano nests that the birds build accrete year upon year upon. And as a result, when they were discovered, quote unquote, by European explorers, there were huge piles of guano. And so here you're seeing a contemporary picture or contemporary etching of one of the Guano Islands, and this huge stack here, this is all the accumulated waste of thousands of years of bird populations living in this area. Well, it was recognized that actually the explorer von Humboldt recognized that this guano material might be an effective fertilizer, and they began sending fertilizer, guano, back to Europe to be used as an agricultural fertilizer. And getting this out of there produced horrible conditions. And this is an example of what the guano mines. And having to work in a guano mine, sounds like a pretty good description to me of hell on earth. Here you are in this, trapped on this little tiny island. There's almost no fresh water because it never rains, and you're having to spend your days, shoveling, this, very powdery bird manure into crates and things, but it was profitable enough, and it was important enough that there was a whole trade of ships that would sail all the way around Cape Horn, all the way to the Guano Islands, fill up with this bird manure, and then sail all the way back around to Europe. And there was sufficient demand and sufficient importance that the trade was sustainable for some period of time. Well, what do cormorants eat, you might ask, to allow them to have such hard populations? Well, they eat a particular fish called the anchovy. And I won't have time to get through all of the material on the anchovy, but the anchovy are a small fish. They're, let's see the picture here. They're, just about a couple inches long. They're a pelagic fish, which means that they live in the surface waters of the ocean. Pelagic means freely swimming typically in surface waters. And they grow very rapidly. So their total lifespan is about 2 years. And so from, larval egg stages to maximum adulthood is a total of about 2 years. That means that the population can grow or shrink very rapidly. Now, why are there so many anchovy? Well, it has to do with upwelling currents. And so in typical situations, there's a south going a current in winds that move along the coast of blow from south to north along the coast of South America. Well, what do we know about wind? Well, we know when wind starts moving ocean waters in the southern hemisphere, Coriolis is going to deflect that current to the left, right? To the right in the northern hemisphere, to the left in the southern hemisphere. So that means that the traditional winds blowing along the coast of Chile are going to drive water offshore in the surface. Well, in order to compensate for the offshore motion of surface waters, there is upwelling from deeper waters. And so there's a constant upwelling of deep ocean water along the coast of South America. And that upwelling of nutrients is what allows populations of anchovy to achieve such high numbers. Okay, so it's end of class. I'm going to have to stop here, but we'll take up our conversation about nutrients from the sea and about the importance of anchovy when we meet again on Thursdays. So I'll stop there. If there are any questions, please ask now, or you can always email us or text us if you have questions about anything going on. Let's see, there's a question in the chat, looks like. Let's see. Okay. All right, with no questions, I'll go ahead and end the class. And so you'll have 2 quizzes this week. There'll be a quiz over the material and nutrients from the sea, and there'll be a quiz, a shorter quiz over the material that I presented on primary production. Both of those quizzes will be in the module for this week. And per request, I'll put in a little example of how you would calculate the intensity of light at some depth, or you could calculate the K coefficient, the coefficient of light extinction, based on information about light at the surface and at depth. Okay, we'll see you all on Thursday. Bye-bye.